Welcome, everybody. Welcome to our webinar. We're going to give people a couple of minutes to log on because we know that all sorts of people from all over the place are going to be logging on. So we're going to give folks a couple of minutes, and then we will start today's webinar. We're very excited. We have two absolutely fantastic speakers, and this is a topic that is certainly very pertinent to NINDS, but I think it applies across a wide variety of other specialties beyond neuroscience as well. So let's see, we're sort of watching the numbers going up. I'm going to wait for the numbers to plateau a little bit, and then we'll go from there. And if you're not speaking, we'd ask that you turn your camera off just for convenience, and obviously your mic as well. Hope everyone's enjoying the hot summer weather. Some places hotter than others. And hopefully nobody was affected by the weather conditions on the West Coast and the events overnight. So our numbers are still going up, which we expect. For those on the East Coast, you're probably hopefully you're not missing too much of noon conference for this one. For those on the West Coast, you're probably starting your workday, or you're already part of the way through your workday. You've probably finished rounds already, your pre-rounds. So we're going to go ahead and get started. So welcome to today's webinar in the series called Challenges in Pediatric Neuroscience Research. My name is Adam Hartman. I'm a program director in the Division of Clinical Research at the National Institute of Neurological Disorders and Stroke, which is one of the institutes at the U.S. National Institutes of Health. I'm also a practicing child neurologist and epileptologist. I have a number of pediatric clinical trials and other human studies in my portfolio. We created the series initially to help investigators overcome some of the challenges they might face in pediatric neuroscience research, including neurology, neurosurgery, psychiatry, psychology, neurodevelopmental disabilities, physiatry, critical care, emergency medicine, pathology, radiology, pharmacology, just to name a few, and there are obviously many others. Our ultimate goal is to improve the quality of care for our patients, but the more proximal goal is to enhance research that will facilitate improvements in practice. Our webinars are targeted to trainees, senior investigators, and even to those who are not currently involved in research. So we're trying to make the pie bigger, so to speak. So today we're hosting Dr. Sunny Juul from Seattle Children's Hospital and Dr. Berry-Kravis from Rush University. This should be a very thought-provoking webinar. A number of very well-done preclinical studies in the space have led to high-profile clinical trials, which ultimately did not meet their primary endpoints. So we're going to explore possible reasons. The topic should be of interest to basic translational and clinical investigators, as well as funders and people with lived experience. Both of our speakers today are the rare breed of basic scientists, translationalists, and clinical trialists. So first, a few technical notes about the webinar. The session is being recorded, will be transcribed and archived on an NINDS website. Details about where to find the archived version will be posted on social media and newsletters. There will be a question and answer segment at the end. Questions can be entered into the chat. I will moderate the question period. Special thanks to Dr. Alvarecinos from NINDS for her technical support and organization in coordinating the webinar, and the NINDS Office of the Scientific Liaison for all its support and publicity. So let's get started. Our first speaker is Dr. Sunny Juul. Dr. Juul is a professor of pediatrics, adjunct professor of neuroscience, director of the Institute on Human Development and Disability, and director of the Intellectual and Developmental Disabilities Research Center at the University of Washington and Seattle Children's Hospital. She was division head of neonatology from 2015 to 2021. Nationally, she's been the chair of the neonatal subboard of the American Board of Pediatrics, a member of the Committee of Fetus and Newborn, and liaison to the American Academy of Pediatrics Executive Committee, a founding member of the Newborn Brain Society, and past chair of the Developmental Brain Disorders Study Section here at NIH. She also is a co-editor of Avery's Diseases of the Newborn. So with that, Dr. Juul, lead us off, please.
Thank you so much. I'm going to go ahead and share my screen here. And thank you for the invitation to talk about this. It's a topic that's near and dear to my heart. So I have nothing to disclose. The preterm EPO neuroprotection trial, or the PNET trial, was supported by NINDS, and in fact, Adam was my program officer. And the use of erythropoietin is off-label for neonates. My objectives are to discuss the background data and rationale for the PNET trial, show you the PNET protocol, the results from the study, and then factors that may affect the outcomes of a clinical study. So just so we all are on the same page, extremely low gestational age neonates, or LGANs, are less than 28 weeks of gestation at birth. There's about 40,000 per year born in the United States, and they have an 18% death rate. 40% of survivors develop severe long-term developmental impairment. And you can see the statistics there, cerebral palsy, intellectual disability, hearing loss, blindness. But also, there's a high percentage of these kids who develop autism or ADHD and later psychiatric problems. So this is a target population that really needs um, uh, help. So we asked the question, can erythropoietin, or EPO, improve these outcomes? And at the time that I was planning this study, there were hundreds of preclinical studies that had already been published, but most of them were in rodents using the Nucci model or middle cerebral artery stroke models. And the results were remarkable. So there was anywhere from 40 to 80% decrease in the size of infarctions. And then work had been done figuring out the appropriate dose and how many doses and so forth. So multiple EPO doses were shown to provide optimal benefit. There were short and long-term studies showing improved outcomes and both histological and behavioral improvements. The neuroprotective dose range was 1,000 to 5,000 units per kilo per dose, given up to 24 hours after injury in rodents. And when we looked at rat versus preterm human study dose comparisons, we identified 1,000 units per kilo as the optimal human dose. So mechanisms of EPO neuroprotection include acute effects as well as long-term effects. So acutely, in these preclinical models, EPO decreases inflammation, decreases apoptosis, and oxidative injury, and that improves net cell survival in the brain. And then long-term, obviously, EPO increases erythropoiesis, which increases oxygen delivery and decreases free iron. It promotes angiogenesis, again, which increases oxygen delivery. And neurogenesis and oligodendrogenesis have been shown in rodent models. So there were phase 2 trials that were done, and you can see some of them here. There was a meta-analysis of these studies that was done. And that basically showed an improvement in cognitive deficits with EPO treatment, although it's interesting that the population and the doses of EPO were quite different in these various studies. And they showed a trend towards a decrease in cerebral palsy. So seemed like a good trial to move forward with a phase 3 study. So we did the PNET trial, which was a randomized controlled phase 3 trial. We enrolled 941 preterm infants that were 24 to 28 weeks. They were enrolled between 2013 and 2016. We had 19 sites and 30 NICUs. Our primary hypothesis was that early high-dose EPO plus maintenance dosing through 32 weeks postmenstrual age would decrease the combined outcome of death or severe neurodevelopmental impairment in LGANs from 40% to 30%, measured at 2 years corrected age. And that if you include moderate or severe neurodevelopmental impairment, that would decrease from 60% to 40%. And death was estimated at 18% based on the current literature at the time. So this was the protocol. We enrolled babies within the first 24 hours of life. Some babies were also enrolled prenatally. So after we obtained consent, we got head ultrasounds and EPO level and circulating biomarkers. The babies got 6 doses of 1,000 units per kilo of EPO or placebo IV. And then for maintenance injections, they got 400 units per kilo of sub-Q EPO or sham injections. So the babies on the placebo arm didn't get shots. 200 babies got MRIs at 36 weeks postnatal, and then we followed the babies up to 2 years of age, and this was our final outcome assessment. So we did a Bailey exam, a standardized neuro exam, and obtained blood pressure, urine, and blood. So here's our consult diagram. We assessed over 3,000 patients, excluded many, and randomized 941. They were pretty well allocated to EPO versus placebo. And we had ultimately about 79% had final outcome data. So we showed that EPO was safe. When we were planning this study, there was a lot of concern about retinopathy of prematurity being increased when EPO was given. That turned out to be not true, as you can see here. And basically, all of the serious adverse events that we looked at were not increased by EPO. We did show that EPO decreased the number of transfusions, cumulative volume, and donor exposures, as you can see here, and also more EPO-treated babies remained transfusion-free, so 139 or 138 versus 59 in the placebo group. But when we looked at our primary outcome of death or severe neurodevelopmental impairment, we did not see what we hope to see, a decrease in death or severe neurodevelopmental impairment. And we looked by gestational age. We did see an increase in benefit at 27 weeks, but there wasn't like a trend by week, so I don't know what that was about. Severe neurodevelopmental impairment trended to be lower. And cerebral palsy was actually lower than expected. But again, these crossed unity, so were not statistically significant. Of note, death and severe neurodevelopmental impairment was lower than predicted. So only 26% of the babies had these outcomes. And so potentially we were underpowered because we did our power calculations based on a 40% prediction of death or neurodevelopmental impairment. And similarly, there was fewer babies with death, moderate, or severe impairment. So why didn't EPO show benefit? Well, you have to ask, what is the relevance of the preclinical data? I mentioned in the background that most of the studies that showed EPO neuroprotection were in rodents. And this is a great example here. There was really robust data in rodents that EPO decreases inflammatory mediators. But you can see here, this is data from peanut. The black shows the placebo kids, the red shows the EPO kids, and there was no difference in any of these tau, IL-8, TNF-alpha, etc. There's no difference in any of the inflammatory mediators in the human children, so unlike rodents. There were also unintended consequences. So, excuse me, the sites were unable to agree on an iron use protocol or a transfusion protocol, even though we tried hard. But people felt really strongly about giving iron, that some people thought that it was dangerous, didn't want to give it. Other people thought iron was very important for brain development and wanted to give it. And we just couldn't agree. And this study was done before the PINT trials and the more definitive transfusion protocol trials. So again, people were either um, low transfusers or high transfusers. So we couldn't come to agreement. We did have guidelines for iron, but you can see here if in this chart, so by 2 weeks of age, the median ferritin in the placebo kids was 164 versus 70 in the EPO kids. And ferritin under 76 is considered mild iron insufficiency. By 4 weeks, the EPO kids were lower. By 12 weeks, severe iron deficiency was noted. And this is showing the ferritin levels, similar results for zinc protoporphyrin. So despite the fact that our EPO-treated kids got higher doses of EPO, of iron, they were insufficient. And we showed that Bailey scale results were better with higher cumulative doses of iron. So the babies who got low doses of iron did worse cognitively, also motor and language performance. So again, that just emphasizes that iron is essential for normal brain development in preterm infants. So there was also site variability that affected outcomes. So you can see here, this is the 19 sites that we had, and this is a transfusion volume. And so there's quite a bit of variability from site to site on how they practiced transfusions. And we showed that transfusions were associated with lower Bailey scale scores for cognitive, motor, and language development. So the more transfusions you got, the worse your Bailey scores were at 2 years of age. And that tended to be more significant for the placebo kids than the EPO kids. Similarly, there's quite a bit of variability for the use of opioids and benzodiazepines. So again, here you can see the 19 sites and the usage of both either or no opioids and benzodiazepines. And here's opioids by themselves and benzodiazepines by themselves. And not surprisingly, we showed that prolonged use of sedatives can worsen outcomes. And so here you see the cognitive, motor, and language scores for babies who got 10 days or more. Outcomes were significantly worse for all parameters. Steroids was another thing that was quite variable depending on site. So this is cumulative steroid dosage versus days treated with steroids, and there's quite a bit of variability by site. And again, we had differences in outcomes based on how many days of steroids babies received. So lessons learned. I think preclinical preliminary data in multiple animal models is critical. Rodents are great, but they're not necessarily reflective of what's happening in human infants. And I think we have several other animal models like piglets and fetal sheep that have gyrified brains, and it's really important to show preliminary results in those animal models as well. And phase 2 studies don't predict the outcome of phase 3 studies. That's why phase 3 studies are so important. Site variability has enormous effects on outcomes, and so it's super important to standardize as much as possible. And there were things that we tried to standardize, like the iron and transfusions. Other things we just didn't even think of standardizing, like some of the other, steroid use, um, um, benzodiazepine use, and so forth. But so really just think about anything that can potentially affect outcomes and standardize as much as you can, and then learn as much from the data that you've collected. So from this peanut trial, we have, oh gosh, 25 or more papers that we've written based on the data that we collected. So thanks to all of the PIs, the research coordinators, our follow-up staff, Adam and Scott Janis, who are our program officers. Jack Widness was our medical monitor, and advisors were Donna Fierro and Roberta Ballard. And of course, our peanut families. So thank you so much.
Dr. Juul, that was fantastic. Our next presenter, so we're going to do, please feel free to enter questions in the Q&A section, the tab up on the top. And we'll do Q&A after Dr. Berry-Kravis's presentation. So I'd like to introduce now Dr. Liz Berry-Kravis, who's a professor in the departments of pediatrics, neurological sciences, anatomy, and cell biology, and the co-director of the molecular diagnostics section of the genetic laboratory at Rush University Hospital. She established the Fragile X Clinic and Research Program in 1991 and has been a leader in translational research in Fragile X for over 20 years, including development of clinical outcome measures and biomarkers, natural history studies, newborn screening, and clinical trials, which we're going to talk about today. Her laboratory studies the cellular role of Fragile X mental retardation protein, FMRP, its relationship between FMRP and clinical function, and optimizing genetic testing methods. More recently, she's expanded her clinical and translational work to other neurodevelopmental disorders as well. She's also on the advisory or review boards for a number of disease-based foundations. Dr. Berry-Kravis.
Thank you, Adam. Okay, I'm going to share, and hopefully that's all going to work. Okay, are we good? I'm going to talk about challenges in Fragile X syndrome, and thanks, Adam, for inviting me to talk about our journey into the valley of clinical trial death in Fragile X, and a little bit about our digging ourselves out of it. These are my disclosures. I work with a lot of companies developing drugs for genetic neurological diseases of children. I don't take any of the money, and it all goes to Rush, where hopefully it supports our rare disease programs. Typically neurodevelopmental disorders have been treated with supportive treatment, but the interest as genes have been identified and the big increase in neuroscience research has occurred over the past 20 years has been to try to switch to the idea of treating these diseases based on their disease mechanism or reversing their genetics. And Fragile X is actually one of the first examples of this. We had the gene back in 1991 and thought, okay, now we're going to find out what the gene does and we can treat the disease based on the genetic effects. So we learned a lot of lessons as the first sort of genetic disease to go down this road for a neurodevelopmental disorder. And a lot of those lessons have been used as other diseases have gone forward to try to do the same thing. FMR1 is the gene defective in Fragile X syndrome. FMR1 normally will have about 10 to 45 repeats. In Fragile X, this is an expanded CGG repeat sequence of over 200. The gene gets methylated. There's no FMR1 RNA, and then reduced FMRP or no FMRP. And it's the absence of FMRP or deficiency of it that leads to the synaptic deficiencies and intellectual compromise. And this disease occurs in about 1 in 4,000 males and females worldwide, is the most common genetic cause of intellectual disability and autism spectrum disorder, and we diagnose it with a DNA test to identify the repeat size. But what does FMRP do if it's so important to cause the disease, it works in an RNA binding complex that, stained here in red, localizes to dendritic spines. You also can find it in axons, but it's best studied in dendrites, where it regulates activation-dependent dendritic translation through multiple signaling pathways. Very complex mechanism. And what we do know is that when FMRP is absent, we get these immature, long, spindly spines in the FMR1 knockout mouse relative to the wild type that has the more mature spines, and that morphological immaturity corresponds to synaptic alterations and deficits, including exaggerated LTD in hippocampus, abnormal LTB in both hippocampus and cortex. And these, the synaptopathy and what exactly it looks like varies by brain area and neuron type. So it's a very complex disorder to think about treating. And the complex sort of brain molecular abnormalities correlate with a fairly complex and variable disorder in terms of severity and manifestations. Most patients will present with developmental delays. Females may present later because this is an X-linked disease, so they're less effective due to their normal X chromosome being expressed in a percentage of their cells. This is a characteristic facial appearance of a boy with Fragile X who actually has all the facial features, like the large ears and the long face. But the physical and medical issues associated with Fragile X are less of a problem than the cognitive deficits and the severe behavioral issues. And those are highly variable. But the cognition is really the core deficit that results in developmental problems and impaired functioning later in life. And this is exacerbated by the behavioral issues, some of which probably come directly from the genetic deficit, and some of which come because of the intellectual disability. And these kind of factors interact to produce dysfunction. Treatment has traditionally been supportive with therapy and behavior plans, educational programs, managing medical issues, and psychopharmacology for behavior. And when we ask how well the supportive behavioral treatment in Fragile X actually work, we've studied that. So this is a within clinic study where if we start patients on these different agents that are matched to whatever symptom they have, we can look at response rates, and these are clinical response rates. So we get a response rate of, up to sort of 70 or 80% if we try multiple different types of medication and optimize the therapy. But these are not normalizing the symptoms. They're improving things a little bit. And Fragile X patients are very hard to treat because they'll have multiple types of limiting behavior, we know from our Forward Natural History Project. And so one medication may aggravate another behavior. Basically, often patients wind up on multiple medications just because their behaviors are so functionally limiting. And then this is a study from Don Bailey, the family perceived efficacy of supportive medications, where the people in green think their medications are working pretty well. The red people think, oh, they're working a little, and the blue people think they're not working at all. And it turns out the only thing we're really good at treating in Fragile X is seizures, and that's because the seizures aren't so severe. So there's a huge unmet need for better behavioral treatments for anything that addresses cognition, and we need to have better supportive medications as well. But it would be ideal to treat the underlying disorder. So we've tried to treat the underlying disorder, and this is just a mechanism slide of Fragile X. So normally FMRP would be sitting on the ribosome regulating all this protein synthesis in this dendrite. But because the FMRP is absent, there's over-signaling through this pathway and overproduction of all these proteins that FMRP regulates. And so we've come up with many ways to try to target this mechanism. We could block down receptor signaling, here shown the mGluR5 receptor that received a lot of attention in Fragile X. We could block the signaling pathway to reduce the overactivity. We could block the proteins that are being made to excess. We could activate AMPA receptors, which are deficient in Fragile X neurons. We could work on GABA to try to decrease this glutamate signaling. We could work directly on proteins using microRNAs, or actually Fragile X regulates a number of individual channels, and we could work on those. So in animal model research, all of these targets produce improvement in the FMR1 knockout mouse. It turns out the mouse is fairly easy to treat. Many have actually been in trials. All of these circled things have been in at least a small Fragile X trial, and many of them have not gone forward either because there was no benefit or because there was no path to direct the drug to. So some of these did get to phase 2, 3 trials and or the phase 3 trials or large phase 2 trials, and those are shown in these big arrows. All the little red arrows are things that couldn't go forward for some reason or another. The green arrows are things that are still being evaluated for Fragile X. So what I'm going to do is try to talk a little bit about the things that got to those phase 3 trials and why they failed. So the first thing to go into trials in Fragile X was baclofen, which is an enantiomer of baclofen, which is used for spasticity. All neurologists know what this is. It was brought into trials by Seaside Therapeutics, a small biotech. We went to the FDA and said, we think this drug works in the animal model to reverse some things in Fragile X. They said, well, they're using the ABC in autism, so we should use that in Fragile X for to measure behavior. And they wanted us to pick one area that would get better. So we had no idea how to do that from our mouse work, but we did pick ABC irritability because that's what was used in the autism trials, and did a placebo-controlled phase 2 trial, which wound up showing that when we did a visual analog scale of the 3 worst behaviors that families were reporting in their child with Fragile X, that showed a statistical improvement, as well as the aberrant behavior checklist, which we had factored for Fragile X since it didn't look like it performed correctly for Fragile X, and had a social avoidance scale that also showed improvement. But there was a trend in all the other measures except the primary outcome of ABC irritability. So we said, well, we picked the wrong outcome. There's enough signal to move forward. We wanted to use this visual analog scale. The FDA didn't like that because they thought the chosen behaviors would be too variable for labeling. And they refused all of our other global measures and insisted on a pediatric multiple dose study with lower doses that weren't therapeutic. So we went into the phase 3 trials with a few things kind of against us. And we ran a trial in 12 to 50-year-olds, which did not show efficacy, a trial in 5 to 11-year-olds, which had to stop early. There was a goal of 200 enrollees, but Seaside went out of business when the trial failed. And ultimately, in the highest dose group of the pediatric trial, we did see a significant benefit in parenting stress and irritability, and almost in the primary outcome of social avoidance. But that had a higher placebo effect because we were enrolling based on having a certain amount of social avoidance, and families were posting online how to score the ABC to get into the trial. So after Seaside went out of business, we looked at 30 individuals in the long-term extension, and it looked like we were normalizing. Normally adaptive scores go down with time in Fragile X because the patients don't keep pace with their typical peers. It looked like that was normalized on our baclofen. And then many years later, Allos came into being and got the phase 3 trial data out of the original trial database, did an analysis and asked, let's look at meaningful change because on the suggestion from the FDA. And they looked at how many patients had meaningful change in these different measures on the aberrant behavior checklist. And so when you look at 3 measures together, the social avoidance, social unresponsiveness, and irritability, it turns out that we had a response for our baclofen in 45% of patients and for placebo in only 4%. And this drug really, having run all these trials, this drug really does work for certain individuals, but not for everyone. And that was highly significant. But the FDA said, well, you need to run a new trial, and we're still working on that 12 years later. Now, okay, not going down. Okay, so now I'm going to shift gears and talk about the mGluR5 blockers in Fragile X. This is an area where the NIH has invested a lot of money. This is the model with the most positive preclinical data possibly ever in a genetic disease. This is the list of mouse phenotypes reversed. You can see there are things from molecular to electrophysiologic to morphologic to behavioral phenotypes. And this is just the seizure phenotype with MPEP showing a reduction of the audiogenic seizures. This is the epileptiform bursts being normalized with MPEP, which is an mGluR5 blocker. And then these are normalization of the dendritic spines. 6 phenotypes also reversed when the FMR1 knockout underwent a genetic reversal by crossing to mGluR5 heterozygous mutants with less mGluR5 receptors. So over 60 papers from 20 labs, many with NIH funding, showed reversal of about 90% of reported mouse, rat, and fly phenotypes. So this was a huge body of support. I mean, why wouldn't you take this into people? As this Fragile X dad said, it's a great time to be a Fragile X mouse. What are you going to do for my kid? So we did go to people, and we ran an early target engagement study with Phenibon, which is an old mGluR5 blocker that showed we could see an improvement of pre-pulse inhibition over test-retest controls. And then Novartis and Roche ran big phase 2b trials in multiple countries, and they chose behavioral outcomes that had huge placebo effects, and they showed no benefit. So Roche and Novartis disbanded their programs, and we asked what went wrong. How did all that preclinical data in 3 species with 4 drugs and genetic and pharmacological correction and target engagement not lead to registration? Well, we had this. So this was the Novartis trial, and there was a placebo run-in, and the placebo change in the placebo run-in was so huge, it makes it almost impossible to see a drug effect. And in fact, in the adult study, the placebo patients, before they were even randomized to drug or placebo, had way more placebo effect than any other group. And then they just kept having more placebo effects. So this became an impossible thing to interpret. And we thought, okay, we ran these trials wrong. We have so much placebo effect that we didn't learn from these trials whether the drugs actually work or not. And we also had other questions like, can you really reverse things in adults? And shouldn't we be studying learning, since Fragile X is a cognitive disorder? And then the mouse work, much of the mouse work was based on learning. And there was a signal in the mouse that younger mice responded better. So we got a big NIH grant. I was the PI of this through Neuronext for a pilot trial to try to create a new paradigm for the development of mechanism-targeted pharmacotherapy in neurodevelopmental disorders and study the effects of the drug on plasticity. I also got an investigator-initiated grant from Novartis to get the drug. And the idea was we would address all 5 problems that we identified in the early mGluR5 negative allosteric modulator trials. So we would use a young age, 3 to 6 years. We would do a long trial to study learning. We would embed a language learning intervention to try to push learning in a specific area so that we could focus the learning on the specific language parameters that had been developed by Len Abaduto, where you can see that just with the intervention, the Fragile X vocabulary goes up. We would have objective language and cognitive measures that weren't going to be subject to so much placebo effect, and an observational videotape communication measure called the weighted communication scale, which would be the primary outcome and would be targeted to the type of language the patients were learning from this the parents gave the language learning intervention in the home. And so they learned it, and then they gave it to the child all the time, so it would rise above any of the other kinds of therapy and things they were getting in the background. And then we had biomarkers of target engagement. And this was the first ever multi-sites trial studying the effects of a targeted drug treatment on learning. So this was the design of it. We had a placebo lead-in for 4 months. We dose escalated to the maximum tolerated dose so that we wouldn't have people on a dose that was too high, and then we're having side effects from that. We randomized them to AFQ, which was the mGluR5 blocker or placebo. And then at the end of 6 months, at the end of 2 months after dose escalation, they started the language intervention. And then at the end of 6 months, on the language intervention as well, we assessed their measures of cognition and language, and then they all had an option to join an open label treatment period. So we enrolled 110 kids to randomize 100 and have 80 finished the placebo-controlled period. That was what we needed. We statistically, we enrolled young kids. English was their primary language. They had a developmental quotient that showed they already had developmental delays, and they had a Fragile X full mutation. And then these were the sites that we used. And then I'm not going to go through all this, but we did a lot of that standardization that was just mentioned. We did a lot of standardization of our measures, our rating. We had central raters. We had a lot of fidelity training, and we really tried to do this trial as tightly as possible. And we can use all of this process we developed for future trials. So what did we see in FX Learn, which was the name of this trial? The study groups were well matched. We had 50 AFQ patients and 49 placebo. We had an even distribution of all the children's characteristic, age, sex, medications, therapy, and the parent characteristics, their sex, education, because the parents were delivering the intervention. Between the groups. We had no placebo effect during the lead-in for either the clinician global impression or the weighted communication scale. And the drug turned out to be very safe. Some kids got activated with a little more insomnia and hyperactivity, and we knew that was an issue from the prior trials. But that suggested that that could abate with time if some of the patients, this kind of went away in the open label extension, or they got titrated down to a dose that didn't cause it. There were no serious adverse events. However, in the primary outcome, there was no difference in the change between groups over the 8 months of drug treatment, placebo-controlled drug treatment in the intent to treat or in the per protocol sensitivity analyses. In the secondary outcomes, there was no difference on any clinical measure, including things like standard developmental measures like the Mullen, the Vineland, MacArthur-Bates, which is a language measure, nor in the blood or the ERP biomarkers. And both groups showed a significant developmental increase in language on multiple measures, which is expected to occur because they were very young children, so they were still developing language. So this study did not show benefit of mGlu-R blockers, even when we ran a very tight study and tried to correct all the things that had been problematic with the original studies from Novartis and Roche. So what did we learn from all of this? Well, from both the Seaside experience and the mGlu-R5 experience, we learned, well, one thing is we need to study the entire disease and not one behavior. There's no one behavior that represents Fragile X. You have to study the whole disease. Behavior may not be a surrogate for cognition, and we need better outcomes of cognition. We need to deal with subgroup effects because we're going to see that with drugs. We have big variability and placebo effects on caregiver questionnaires, especially if we run this in multiple countries and cultures, and if males and females are in the study. This makes studies uninterpretable. We need more objective outcomes, and we need to narrow down our groups a little bit more and standardize things better. The FDA is often not right with rare diseases, and we need to argue with them on some kinds of things. Small companies depending on venture capital should not try to do too much, because when Seaside tried to go into autism, that was when they ran out of money and had to close for Fragile X. You should focus and get the primary thing approved. This also prevented trofinetide from going forward in Fragile X because Acadia did the right thing and just focused on Rett syndrome. Don't enroll based on the primary outcome. Do not let families post stuff on social media. That really caught us by surprise back in the 2008, 2009. And then most importantly, from all the mGlu-R5 work, mouse findings may not translate to humans at all. The mechanism may have evolved apart. Some pathways like mGlu-R5 may be different or less critical or linked to different outputs in neural cells in man relative to mouse. And some pathway targets are going to translate, but we have no way to predict our priority, which ones. So we need to measure a phenotype in the animal model, like a biomarker, like an EEG measure that can be directly translated, and you can see the same thing in man, so you know the abnormality is present in both species. And we did not have that for the mGlu-R5 negative allosteric modulators. And so we also need to do early, high-intensity, small, fast-fail studies to see if we can see a signal across species, and don't go forward, and this study is not positive. Weed out those where the pathway target has evolved and does not translate before putting resources into a big trial. And the NIH has actually changed funding priorities, I think, in the Fragile X centers to reflect the above, so that they're really trying to look for ways to go across from mouse to man. Okay, and so what we did in the Fragile X field is we created the Clinical Trials Committee, and companies cannot do a trial in Fragile X without talking to us. We provide guidance. We want to avoid the past mistakes. We talk to them about trial design and outcomes, preclinical and clinical development, and the National Fragile X Foundation will not endorse a trial unless the Clinical Trials Committee has said this looks like it's going to be well done. And so how did we apply these learnings? Just a couple slides. We developed more appropriate outcome measures. We developed these EEG ERP findings that translate directly from mouse to man. And we looked at how do we look at personalized ratings of the most problematic symptoms, since Fragile X patients differ from one to another. We validated the NIH Toolbox Cognitive Battery work of David Hessel for intellectual disability. And then the next targeted treatment that had a drug ready for it was cyclic AMP deficiency, which I actually discovered in 1990, and then was shown to be present in the mouse and shown to cross species, which is really important, again, to see that the mechanism is crossing species. The mouse, Drosophila, and man all show the same thing. And so we were awaiting a drug with less off-target body effects, and in 2017, BPN-14770, the phosphodiesterase 4D inhibitor, came from Tetra, and that enzyme is mainly in brain, so it doesn't cause the off-target body effects. They were able to show that it reversed behavioral phenotypes in mouse, but more importantly, it reversed the cyclic AMP deficit in mouse and had a 14-day carryover. So we designed a trial, which would be one of those fast fail trials. We're not going to do a big trial unless this trial shows us a pretty significant signal. And so we had a crossover trial to look and see if we could see the carryover effects in human that we saw in mouse. And we incorporated the new, better clinical outcomes with the NIH Toolbox, the personalized scale, visual analog scale, and the biomarker ERP, as well as eye tracking. The carryover, there was a carryover effect in humans, so we did all analyses on the first treatment period. There were no safety issues and no GI problems. And all 4 NIH Toolbox cognitive battery measures, I nearly fell off my chair when I saw this, but in 15 patients on drug versus 15 placebo, all 4 of these NIH Toolbox cognitive battery measures improved. And there was improvement in the crystallized composite, which combines the picture vocabulary and oral reading as a measure of verbal cognition. The caregiver rated most problematic language and daily function items, averaged a 15-point improvement in language and daily function. And these were just some of the items that the families picked to rate during this trial. And they were the items the families felt were problematic for that individual. And then there were trends on most other clinical measures. The EEG showed a drug-related decrease in the abnormally large N1 on ERP that actually correlated with the PK in the blood. And then we saw improvement in the abnormal alpha peak as well. So we had EEG findings. And this was really the first trial ever in Fragile X. We had improvement on an objective cognitive measure, parent ratings, and an EEG biomarker. So we've gone on to phase 3, and those are near complete, and we'll find out this fall whether we have a successful model for how to do trials in Fragile X to try to dig ourselves out of our clinical trial valley. And one of the things about the phase 3 trials that Tetra and Shionogi argued with the FDA to use the new measures in phase 3 with additional validation required. And so if those outcomes are successful and can be validated, they will really help going forward with a bunch of other medications that target different things in the Fragile X pathway, gene therapies, antisense oligonucleotide therapies, FMRP mini protein replacement, which are all headed for trials in Fragile X. And so that's all I'm going to say. Hopefully I left time for questions. And this is just all of our many collaborators over the year, our industry partners. We've had funding from all the federal agencies and have trained many people. And then the Fragile X families and patients who are really our partners in the journey to develop new treatments that will hopefully improve quality of life. And so I'm going to stop sharing. And
yes. So hopefully Dr. Juul can come back on as well on video. Those were 2 absolutely fantastic talks. I know we've kept putting this webinar off. We actually had planned to do this a lot earlier in the year. And for a variety of reasons internal to NIH, we had to postpone it. But I'll tell you, one of my colleagues said internally, this is totally worth the wait. This is just, both of you were just absolutely phenomenal. So we're going to start the question and answer period. It's going to be a little bit short, but that's okay. We're going to end at 5 minutes before the hour to respect everyone's time. And, we're all sort of bouncing from webinar to webinar. We're going to have more time, more questions and time to answer. So if you do have a question that wasn't asked, that we're unable to ask, send it to us, either to the presenters or to me, and then we'll get it answered. So as a reminder, you can use the Q&A tab at the top of the screen. We're not going to be doing video questions, live video. We're just going to do the written ones. Okay, so first question, I'm going to use the convener's prerogative. So this is a question for both of you. So you both talked about different animal models. Sunny, at one point you mentioned large animal models, and Liz, you talked about a variety of animal models. So, if you have a common disease where something is an induced factor, that's one thing. But if you have a rare disease that's genetic or rare environmental exposure, it's a little bit harder to do that sort of thing. So in that scenario, do we say that biomarkers, Liz, you kind of alluded to this. Do we say that biomarkers you have to have a biomarker before you're going to do a later phase clinical trial? Or how do we handle the situation where it's really hard, either there isn't a good animal model or it's really hard to develop an animal model before moving something that seems to work in small animals to human beings? So, Sunny, we'll give Liz a chance to catch her breath. Sunny, your thoughts on that?
Yeah, I mean, for rare diseases, that's a much harder issue, I think, because the mouse models are so perfect for that, right? But I think people are working to create models of genetic diseases in other animals like ferrets. But I just, I loved your quote, Liz, about, it's a great time to be a mouse with Fragile X. We can cure almost anything, I feel like, in rodents, and it does not translate. And so um, I just have sort of lost faith, actually, in rodent models. And we've developed a ferret model of brain injury in our lab that we've been using because it's the smallest gyrified brain mammal. And so we're hoping that translates better. But yeah, I don't know. They're all models, right? Like they model certain things, and it may not be the right thing. Just like you were saying, Liz, is like, you test your outcome is one measure. Oh, maybe it's the wrong measure. But I think it's really important to make sure that the changes that you see in your model are reflected in the humans. And I think in my studies, the example of inflammatory mediators not being different in humans treated with EPO was huge, right? And we did that for peanut and in the HEAL trial, the term infants with hypoxic ischemic encephalopathy, and there were no differences in the biomarkers there either. So I think you need to do that homework before you get started in human trials.
Liz?
Yeah, and that's what we're telling all the companies. Our theory is that basic electrophysiologic mechanisms like EEG changes will translate better from mouse to man because all those behaviors are really hard to know what they represent. And so that's what we're trying to do now is show that, or some kind of biochemistry or molecular mechanism that we can see it in humans and we can see it in the mouse. And then we're going to go try to fix it. And we're going to do an early trial that's going to show us if we fix it in the mouse, can we fix that underlying biochemistry or electrophysiology in the human before we try to move to doing big trials. And so that's the goal. But I will say, I think there's a role for iPS cells and organoids also to, move kind of from the mouse into a human tissue, which granted is not wired like brain. So then you lose some of the sort of network effects in developmental disorders. So it may be that we need a combination of things if we really don't have the resources to make a large animal model for every one of these genetic conditions.
Great. Okay, so given the time, we have a short question for one for each of you, and then we'll wrap things up. And so this one I think is for you, Sunny. What materials did you use for language assessment, and what age range was it done?
We used the Bailey scales of infant and toddler development, and we did it between 22 and 26 months corrected age. So 2-year outcome.
Great. And then for Liz, you mentioned that our baclofen works very well in some, but not all people. Were you able to tease out any variables that predicted, or at least associated with, which patients would respond?
Yeah, we don't know that, and none of the biomarker work that Seaside was going to do got done because they went out of business. But there is a suggestion now that the better responders might be those who have more gamma power on the EEG, which is one of the EEG biomarkers for Fragile X, and we'd love to test that out, but there haven't been sufficient funds to go back and do a trial. But that might be a way of actually enrolling, of saying, if you have this abnormality, you're more likely to respond. And so then we can kind of stack our trial with the people who will respond, which is about, 45 to 50% of Fragile X. So it would be a very significant benefit to be able to treat those people. So yeah, more work on that hopefully coming.
Fantastic. So I want to issue a mea culpa. The new name since 2022 for FMRP is Fragile X messenger ribonucleoprotein 1. So I said that incorrectly earlier. So I wanted to thank both of you. This was absolutely phenomenal. Unfortunately, like I said, we ran out of time. We will take the questions that were not asked and forward them to both of you, and hopefully get back to the people who asked them. This is really great. So the session is being recorded. It's going to be transcribed and archived on an NINDS website, hopefully in the near future. And so we'll send everybody who attended, who registered actually, information about where to find that, and we'll also be posting it on social media and various professional society newsletters. So again, thanks a million for all of our attendees. We actually had a great, we'll tell Sunny and Liz later what the attendance was. It was fantastic. In terms of numbers, and the questions were really good, very thoughtful questions too. And to both of you, really, just really thoughtful analyses. I imagine we could have done a whole symposium on this and still have many more questions that are raised, which is the mark of great speakers and great science. So thanks again to both of you. Thanks to everyone for attending today. And with that, we'll wrap up. Thanks again.
Thanks so much.
Have a great week,
Thank you.
everybody.
Thank you, Adam. Bye.